Okay, hello everyone. People are just coming into the session now, so we can slowly get started with our first speaker, uh, Dr. I believe it's going to be Pedro Antunes. I have here. Dr. Pedro Antunes, come on the stage. Thank you. So as we're getting started, I just wanted to introduce myself as the moderator for the next three speakers. So my name is Vicky, and I also wanted to thank our sponsors, our gold sponsors, the City of Toronto, and silver sponsors, the Invasive Species Center, Leuschner's Vegetation Management, Toronto and Region Conservation Authority, and CPRO, and also our bronze sponsors, BASF and Corteva Agriscience. So our first presentation will be Dr. Pedro Antunes on constructing standard invasion curves from herbarium data towards increased increased predictability of plant invasions. And Dr. Pedro Madeira Antunes is a Portuguese-Canadian biologist. He studies the intricate world beneath our feet where soil, plants, and microbes interact, shaping communities and ecosystems. More specifically, he studies the life-supporting role of soil, exploring community-level interactions between plants and soil organisms, with an emphasis on mycorrhizal fungi, which act as conduits of nutrients to plants and carbon in the soil food web. When he accepted a position of research chair in invasive species biology at Algoma University in 2010, and then established the plant and soil ecology lab dedicated to understanding the ecological impacts of invasive species and other environmental stressors. So welcome, Dr. Pedro, and you can share your presentation.
Thank you. Okay, I'll share my screen here and start sharing my presentation. Can you see my presentation?
Yeah, that's great.
Okay, great. All right. So, yeah, thank you for the invitation. And today I will speak about whether or not we can increase predictability in plant invasions and by using trait data, herbarium data. So when an exotic plant is introduced to a new environment, it doesn't usually become invasive immediately. In fact, it can take many years, even decades, before its population grows and it spreads enough to be considered invasive. So in this image, you can see a phylogenetic tree of all the plant species that are present in an old field in southern Ontario, and the ones in orange are not native. So this delay got me thinking a few years ago that if we could better understand this lag time between the time a plant is introduced until it becomes invasive for each exotic species. Maybe if we started doing that and analyzing how long it takes for this transition to occur from being non-invasive to invasive, then cross-referencing that with specific traits of the species, maybe we could significantly improve our ability to predict future invasions. So this would help us identify non-native species that are already present in our ecosystems that have the potential to become invasive. So basically giving us this window of opportunity to be or act more proactively. So together with a colleague of mine here at the Department of Biology at Algoma University, we published a paper exploring how this could actually potentially be done. And this is what I'm going to go through with everyone here right now. Okay, so if we're standing at the front lines of managing invasives, early detection, then the rapid response is our best defenses, the best defenses we have against potential invasive species. But these critical moments, often they go unnoticed. And this is largely because our ability to predict which non-native species will become invasive is very limited. We lack the tools necessary for accurate foresight. So invasion curves, which track the abundance of non-native species over time, and I'll show you one next, they offer this window to understand this process. These curves highlight a crucial period. So the lag phase, when a species exists but hasn't yet begun to spread uncontrollably, let's say. So this phase is basically our golden opportunity for targeted intervention, when we can act before damage, ecosystem damage, other types of damage, societal, economic, etc., begins. So to build effective prediction tools, we need data that is both accessible and standardized. And as I will try to show, herbarium collections are a treasure trove of historical plant records, and they hold untapped potential for constructing these invasion curves. So this graph illustrates the invasion curves for two hypothetical species. So you have species A and species B, highlighting how their invasion dynamics differ over time. So on the y-axis, we have the normalized area invaded, representing the proportion of the total affected area by each species. And the x-axis shows time in years, so indicating how the invasion progresses. So species A, shown in yellow, has a longer lag phase, meaning it takes a significant amount of time before it starts spreading rapidly, but species B, here is shown in orange, has a shorter lag time. So beginning its rapid spread obviously much earlier. So the dashed and the dotted vertical lines, they are the inflection points for both species. So these are those critical moments when the invasion shifts from slow to gradual and then rapid expansion. So recognizing this lag phase is crucial because, again, interventions, they are going to be much more effective and cost efficient at this particular tipping point. So the comparison here demonstrates basically the importance of understanding lag phases and then invasion patterns. So for species with longer lag phases like species A, we have more time to act, and obviously, so the ones like species B, we need to much faster response. So what we have to do here is to extract data from herbarium records to make these curves, right? So actually gather all the data that is necessary for all these different species, native and non-native, and the same ecosystems. And we need to include as many species as possible and create and start extracting this data. And so eventually we will be able to have things like this. This histogram illustrates the distribution of lag phase durations across different hypothetical species. So on the x-axis, we have the lag phase, so duration in years, and so how long, again, a species takes to begin spreading significantly after its introduction. And on the y-axis, we see the frequency or the number of species that fall into each lag phase duration category. So what this distribution shows us is the variation in lag phases. So while some species have very short lag phases under 10 years, others will have decades before becoming invasive. So most species, they will cluster around 20 to 40 years, hypothetically. So then we could start seeing how this is occurring for lots of different species and ecosystems. So imagine if we could generate this type of data for all these variety of species. So then we could combine a fuller picture with additional information like species traits, environmental factors, introduction pathways. We could start identifying patterns and correlations. For example, we might uncover that certain plant traits such as growth rate or seed dispersal mechanisms are linked to shorter or longer lag phases. So it's this kind of insight that could be, I think, if we could pull it together, eventually potential with a lot more computer capacity, could potentially be a game changer for invasion science. So predicting the behavior of non-native species already present, and then take these proactive measures. So the objectives that we had in that paper was to develop standardized invasion curves for extant native and non-native, by non-native, both invasive and naturalized species using herbarium data. And then also to, in what we looked at was trying to address challenges in producing reliable and comparable curves, potentially by thinking about how herbaria can in many ways change practices or adjust practices or sampling efforts. So a herbarium specimen is here, and it consists of a pressed or a dried plant that is mounted, and then it has a whole bunch of data that is collected, where it was collected and what year, all of those things. So data collection from herbarium records also bring a lot of supplementary sources, right? So identification of key phases of invasion curves, and then reaching the plateau is what we would be able to do with lots of looking at a lot of these specimens year over year across different herbaria. So and also a methodology we used in our paper was analyzing biases, standardizations challenges, if we are to start using this data for the purpose of developing these curves. So integration also of spatial and temporal patterns into standardized models is something we looked into. So herbarium data includes plant traits, environmental details, helps us link specific characteristics to invasion patterns. So with these insights, we can develop predictive models to identify non-native species that may still be in their lag phase, right? So as I mentioned before. So what challenges then will we have to overcome? So there's obviously going to be variability in data collection, methods and intensity. So these need to be adjusted. Herbaria talking to a network, so communication and establishment of research goals towards these kinds of efforts. Biases in herbarium data due to geographical temporal inconsistencies will have to be addressed as well. And there are difficulties in standardizing invasion curves across species and regions, and I can talk a little bit about that. And so from there, we came with a bunch of recommendations. So one of the recommendations is to enhance systematic collection of herbarium data across regions. So every year going out and using systematic approaches, looking at all the species that are present, native species, non-native, and standardizing that, and also thinking about at what scale. And when we actually suggest a few the scale that could potentially make this doable. And another thing that could be used is proportion curves. Proportion curves, there are these analytical tools that used in ecological studies. And I'll provide an example here. This graph shows proportion curves comparing the cumulative records of non-native species in red to native species in orange, with a baseline of equal proportions that are represented by the dotted gray line, sorry. So these curves help us better understand and compare how non-native species accumulate records over time relative to the native species. So the native species curve, the dashed orange, serves as a baseline, showing how records. So sorry. Oh,
just another
showing
minute.
how. Yeah. So. And in contrast, the non-native species curve reflects their slower initial accumulated, but then steeply increasing. So why are these invasion curves useful? Allows us to compare across species and regions. We can identify these key phases and then target interventions, and it offers a standardized framework as well. So Also, we can integrate molecular and genetic data for better insights and adopt a standardized scale that I mentioned, say a 10 by 10 kilometer grids for comparisons. Here we're looking at a critical comparison of lag times, and this is just to give you an example. So if I walk through this chart, on the left, we have lag times in years shown by the bars, and then the green bars represent the known invaders, and the orange bar highlights a potential invader. So what are the key observations here? So species like C and E have shorter lag times, meaning they didn't wait long after introduction before they began to spread. So combined with their moderate to high growth rates, these species just became quickly invader. Then we have the potential invader. And here we can look at the relationship between lag time and then growth rate, right? So if you look at the red dashed line, it suggests an inverse relationship, right? Species with shorter lag times tend to have higher growth rates. So this pattern can help us identify which species might invade sooner and also more aggressively. So how does this help us predict invasions in the future? If we can do this for many, many different traits, as I mentioned, this kind of exercise. So in conclusion, standardized invasion curves may enhance our ability to predict and manage invasive species. Alternatively, the random, and that's always something I debate, alternatively, random, very unique traits may hinder invasions actually unpredictable, which is something that I think about a lot. And collaboration across herbaria, as I mentioned, and networks is essential. There's less and less funding for herbaria. Unfortunately, they are a treasure trove. We really need to fund the herbaria more and support them more as a society. And future work should focus on refining methods, integrating diverse data sources. And that's it. I will not talk about this work on traits and pathogen accumulation also leading to potentially predictions about what species may accumulate pathogens faster and decline. Yeah, thank you.
Thank you very much, Pedro. That was very interesting. And if people can access this article, if they want to learn more or get in touch with you, we have a question here for you. Is use of herbarium specimens more or less equivalent to iNaturalist or similar, apart from the issue of proper identification?
Well, yeah, iNaturalist, it would be in some ways similar, as the only difference is that iNaturalist there's a one-stop shop. I don't know how easy it is to download data, access data. I think what I propose here is a framework that would require a lot of funding. It's an idea, and it would need a lot more consideration before it could become usable, and a lot more computer capacity. Maybe in the future with more and more AI and quantum computing, and we could cross-reference all these different traits, ecosystems, herbarium data, but we're not there yet.
Thank you. I think that was all the questions, unless anyone has any more. Can begin to start with our next speaker.
Thank you.
Thank you very much. Okay, so the other concurrent session is just wrapping up the last question.
You can see my slides, right?
Yes, I see your slide. Looks great. So this next presentation is Dr. Richard Hamlin. He's a professor and head Department of Forest and Conservation Sciences for Faculty of Forestry at the University of British Columbia. Dr. Richard Hamlin obtained a BSc from McGill University, Masters of Pest Management from Simon Fraser University, and a PhD from the University of Kentucky. He works on pressing forest health issues such as invasive pathogens and pests and climate change. He has developed tools for pathogen detection and novel genomic approaches for pathogen surveillance. He has trained 60 graduate students postgraduate fellows, and research staff, published 200 peer-reviewed scientific articles, and delivered more than 300 presentations around the world. He was president of the Canadian Phytopathological Society and the Quebec Society for Plant Protection. He is a fellow of the American Phytopathological Society and received the International Union of Forest Health Organization Scientific Achievement Award, the Elizabeth II Diamond Jubilee Award, the Canadian Institute of Forestry Scientific Achievement Award and Merit Award from Natural Resources Canada, the Canadian Forest Service, the Canadian Food Inspection Agency, and the Quebec Society for Plant Protection.
Well, thank you very much, Vicky, and hi everyone. Pleasure to be here talking to you today about genomic biosurveillance of forest invasive species. Now, this topic is going to be a little bit different because I don't work on invasive plants as such, but I work on pests and pathogens that are invasive. And I think this is going to be relevant. You'll see, you know, as I explain, I think it's going to be very relevant to invasive plants as well. Of course, we live in the age of the Anthropocene, so forests and other ecosystems are facing serious challenges. The climate's changing, globalization, you know, the planet has become very small in some ways, and we are facing invasive species, increasing threats from invasive species. In particular, marine transport, you know, every day, every hour, every day, goods are moving around from continent to continent, country to country. Air travel also, of course, you can get in a plane in the morning and go halfway around the world within a few hours. So this has made the planet much smaller, and it has also generated opportunities for, of course, invasive species to move around. Now, I know that this meeting mostly talks about plants, but pathogens and insects move around as well, thanks to, you know, globalization. But in maybe in somewhat different ways. One of the ways, of course, is through wood products and also wood packing material. That's one way that pests and pathogens can move around. All of these containers, all this shipping, all the material that we ship around has to come into boxes. They have to sit on crates, and this is a big source of invasive species. Although there's some mitigation approaches now. Not going to get into that, but certainly we do a lot to try to prevent that. But the other big source of invasive, especially the pathogens, is plants that we move around in the nursery industry, in other, you know, reforestation programs and so on. So we move a lot of plants around. And of course, you look at this nursery and everything looks perfectly green, but we know that, you know, inside some of those plant tissues, the roots, the stems, the leaves, there could be, you know, microbes or you know, that are not, you know, showing pathogenic activity yet. They may be just dormant, but certainly the potential is there for them to come out and wreak havoc on ecosystem. Of course, we have, you know, we all know about this invasion curve, but I just put it up here to emphasize how, you know, the sort of the jackpot is here, the prevention and eradication, and in some ways in containment. But of course, the cost balloon over time as we kind of lose the battle, and then we have to go into controlling and long-term management. So, and this is where, you know, we can do more, especially in terms of our using or developing and using some of the genomic tools that I'm interested in. Because, you know, the story of invasive species is written in their DNA. You know, the sources of invasion, origins, and so on can be obtained from sequencing the genomes of some of those invasive species. And so we developed this area of genomic biosurveillance to enhance this early detection also the accurate identification and the tracking of pathways. So this is one of the approaches that is really complementary with what's already being done, but generates extra data, extra information. So we launched this project BioSafe, Biosurveillance of Alien Forest Enemies, where we aim to generate these genomics resources, and then develop and validate tools to identify forest invasive species, detect them, and also track them and assess the risk that they pose. So we developed this pipeline for biosurveillance of invasive pests and pathogens. We purposefully, target pest and pathogen, not just pest or pathogen, just to show that it's widely applicable. And that's what I'm thinking. This would be applicable, of course, to invasive plants as well. The idea is to develop genomic resources. Of course, we need to generate sort of the database, the standards from global collections. The second step in the pipeline is to identify markers that are useful at whatever level it is that we are interested in. And then design, develop and then validate these tools for genomic biosurveillance so that they eventually can be transferred to users. So whether they're in regulatory agencies or NGOs and so on and so forth, even the industry, can make use of these tools. So the first step, of course, is assembling these global collection of samples. We need to generate the genomic data that can be used so that we can start characterizing, the different species, the different populations. And in the BioSafe project, we did that with multiple pests and pathogens, including spongy moth, Asian longhorn beetle, Dutch elm disease, and some of the pathogens, soil pathogens like the sudden oak death pathogen. And we, out of these collections, we generated genomic data so that we can then look into markers that are specific to geographic origin or to traits, and then identify variants, lineages, and then even hybrids between different species. Now, a little surprise, of course, when we started sequencing genomes in this project with fungi, I had a lot of experience sequencing fungal genomes, and I knew that they were tractable. Fungal genomes are relatively small. If we're talking about the base pairs or the A, Cs, and Ts that are present in a genome, there's about 30 million of them in a fungal genome. By comparison, in the insects, so this is spongy moth genome that we sequence as part of the BioSafe project, we're pretty close to 1 billion. So 30 times higher in terms of the sequencing. So that kind of complicates things. Now, by comparison, the human genome has 3 billion base pairs, so nucleotides. So we're still below that in terms of the complexity. But we think plants could be kind of all over the place. So we actually started a project on an invasive mistletoe species. And the genome that we're looking at is about 9 billion nucleotides. So much, much bigger, almost 10 times as big as our spongy moth genome. So something to be aware of when we start generating those genomic resources. Now, the next step, of course, is to go back and then sample broadly the populations around the world globally. And we did this here on the spongy moth. This is a project that Sandrine Pic had worked on in the BioSafe. Sandrine was a postdoc fellow in the BioSafe project, and she's now a research scientist at Canadian Forest Service. And Sandrine has assembled a fantastic collection of spongy moth samples from around the world. And then, we proceeded to sequence the genome and then obtain genomic profiles And the results are astonishing, really. If you look at the map of the world, this is the actual geographic map, and you can see the different locations here. And at the bottom here is a principal component analysis that uses only the genetic data. There's no geography here. This is just genetic data. And look at this. It really mirrors the map. So this was kind of a really, really cool result for us, because it means that if you get an unknown sample, if you capture an unknown sample on a ship or on a truck, you can sequence or obtain markers, genomic data, and then you can probably easily assign it to a source just based on the genomic profile. So this was exciting results for us, and we continued this experiment by obtaining samples from shippings, from cargoes, and then assigning them to sources. And this was very successful. We found that most of the captured or the intercepted samples originated from Japan, with a smaller number from Russia, some from China, and then the smallest number from Korea. So really informative in terms of where do those samples come from, where do they originate. This is the great starting point to start doing something about it, trying to prevent, further spread, and trying to go back to the source. Now, all of this that I'm talking to you about, involves a lot of markers, basically kind of a big genomic scan, but in a sort of operational way. This is too much. Like, we don't want to have to sequence these billions of nucleotides every time we find a sample. So we wanted to know, can we reduce this and can use a more targeted approach to get the same amount of information. So we started kind of reducing the genome by enriching the targets that we want to work on. And we found that you can go down to the maybe 300 markers and still obtain the same, very similar level of accuracy in terms of assignment of a sample to an unknown. So this was very encouraging to us to know that, we don't have to sequence entire genomes. We can actually target the areas in the genome that we are interested in, and then focus our analyses on those areas. And then we developed a bunch of tools, actually, not just one, but a bunch of tools. The one for spongy moth we call SpongySeq, for spongy sequencing, but we also have some for Asian longhorn beetle called BeetleSeq. We have some for the Dutch elm disease called DeadSeq. The idea being that for each of these invasive species, then we develop, sets of markers that we use to sequence and obtain information that can be then used to provide information on the, taxonomy, species, subspecies level, whether they're hybridized, geography, where do they originate from. And we even have some instances where we can actually have markers for traits. For example, spongy moth, capacity to fly or not to fly. And then, of course, you can, use these tools in surveys, where you can take your samples and obtain some genetic data using these tools. Now, currently, we're doing this in the lab because of the technology that we're using, but it certainly provides a rapid and cheap genome analysis. The extractions are kind of straightforward, easy, and you can obtain a genomic profile for about $30 a sample, which is relatively cheap. And these costs, of course, will be going down. So really ideal for these kind of high throughput surveys, and even for kind of operational identification of origin of samples in points of entry. And the analysis is relatively simple. Just because of that targeting and simplification, we can actually do very rapid analyses that can be done on a laptop. You don't need a mega server to do the analyses. But of course, so this is working now. But the goal is bring it to the field. That's been the holy grail that I've been pursuing all my entire career, is is to try to bring these tools to the field. And we've successfully achieved that with PCR. So taking some of those markers and then developing PCR assays that can be done on site. And this is me with my lab bench, which is a Douglas fir stump in the forest with all of the kits that I need to identify the spongy mud flight capacity, and so that can be done quickly in the field. And the next step, which is completely achievable, is to be able to then obtain entire genomes, or at least a genome targeted approach that we're using, those spongy seq, for example, or dead seq that we could do in the field. So that's kind of the next step in this project. And so this is, I think, like I said, entirely applicable to plants, given that plants, of course, have larger genomes. It might be a little bit more complicated, but certainly the principle of identification, assigning to sources, and then tracking pathways, it's entirely applicable. So this is all I had for you today. Thank you very much for your attention. And this is a view from up in the North Shore Mountain in Vancouver, where I do a lot of my hiking. And it was a pleasure to talk to you, and be happy to answer any questions.
Beautiful landscape and great presentation, Richard. Thank you very much. It was beautiful to hike there. I don't see any questions for you at the moment. We just got a comment, really interesting. Yeah, so there's, I don't know if this is a bit too basic. I'm just wondering, like, why is a moth genome so much larger than a human's?
So that's a great question. We spend a lot of time kind of thinking about that. Why is, for example, a fungus genome is 30 times smaller than a moth genome? So a lot of this is due to what we call repetitive sequences. So we used to call them junk DNA. It's basically DNA that's kind of repeated in the genome. We don't know what it does. Used to call it junk DNA. Of course, it's not junk. It's probably doing something, but we're still not quite sure what it does. The reality is that the number of genes, when we focus on the genes in the genome, it's about the same. So a moth and a fungus and human all have about the same number of genes. And so that's the rest is all, this size of the genome then has to do more with these pieces of DNA that are not genes, but they probably do something else, but still a bit of a mystery.
Thank you. Very interesting. And two questions have popped up. One is, what is the timeline before this becomes commercially available?
So the like spongy seq, for example, that what we've done is that we have an agreement with a company that can provide these kits that can be ordered. But you still need to have the expertise to do the analysis. But we also kind of offer like fee for service for doing it. Now the PCR, there's increasingly more and more portable PCR platforms that can be used. So that's it's just a question of of putting the kits together, and if there's a demand, if there's a commercial demand, it would certainly be easy to develop a product that could be sold commercially.
Another question, merci. For the forest pest detection using DNA device, does it only detect presence, or is it possible to also get a sense of extent of the infestation?
So far, it detects the presence of the organism. In terms of the extent of infestation, that relies on doing the surveys. Now we can kind of approximate, for example, the size of a population using the genomic data. So a very small population, we'd have what we call a genetic bottleneck. There will be way less diversity in a small invasive population than in an expensive population where we find more diversity. So we can sort of guess some of the population size just from the genomic data, but for like sort of the geographic placement, then we need to sample from different areas and then use the DNA approaches to kind of map out where the invasive species are.
Excellent. Great. Thank you, Richard. Thank you for your time. Much appreciated.
Everyone, it's great talking to you.
Mhm.